Thanks for joining us again today, and welcome to the 7th webinar in the Keeping It Real series. So I'm Anna Richards, Commercial Lead at Vitaccess, and I will be moderating today. I'm joined by Dr. Catherine Bottomley, Chief Scientific Officer, and Sam Llewellyn, Senior Consultant. So move to the next slide. Over the next 30 minutes, we will take you through diagnosis validation in patient-centered research. And on the agenda today, we will be going through the overview of Vitaccess through to a background to diagnosis validation, sources of diagnosis validation, and then we move on to the sort of hints and tips section where we will take you through strategies for validating diagnosis. This is all followed by a Q&A section where you can put forward your questions to us using the Q&A box on the side panel. If you have any questions, just please feel free to add them at any point during the webinar, ready for the QA. And I know a few of you sent some in advance as well. So now to give you a quick overview about Vitaccess. So we are an innovative research organisation with these 5 complementary core capabilities. So this allows us really to give a collaborative approach when we're conducting global digital research. Today we have the leaders here of the patient-centered outcomes team to give you their insights in patient validation. So we're focusing on that particularly today. On the next slide, we show you from really the real-world evidence point of view of from concept to implementation, how we help generate real-world data and insights through creative patient-centric solutions. We do this through the Vitaccess Real platform. So that comprises a data capture tool and a real-time research portal that you can see here. So you can see the data as it's collected. On the next slide, you'll actually see the flow from the type of data, the participant data capture. So this could be patients, caregivers, or clinicians that can be captured, and through a omni-channel recruitment, which the guys will take you through in a bit more detail in a moment. But now I will pass over to Catherine, who will take you through the background to diagnosis validation.
Thank you very much, Anna. Hello, everybody. Good morning, good afternoon. Before we launch into the main webinar content, I just wanted to highlight some of the key references that have contributed to our presentation today. So firstly, 2 posts that Vitaccess have produced. So one is a blog post available on our website. The second is a preprint for a conference abstract that we have coming up later in the year. And we've also put together some content from publications from global perspectives who we work closely with in many of our studies. So just to give you a bit of a background as to some of the methods that we use here at Vitaccess for recruiting participants to our studies. So our participants can be patients, they could be caregivers, or they could be clinicians, either contributing data from their perspective or contributing clinical data to our projects. And we use a variety of different recruitment methods depending on the study that we're undertaking. So the first method that we use is clinical sites, and whether that is from a traditional real-world evidence perspective where we are relying on the sites to provide us with clinical data, which we will then align with patient-reported data in the majority of our studies, or whether those clinical sites are being used purely to identify and recruit patients to our studies. We very commonly work with what we call community recruitment. So that is partnering with patient groups, patient associations, very collaboratively from the start of our project right through to the end. And the community recruitment tends to be through methods such as advertising studies on websites, sending out email, newsletters, social media posts, word of mouth. There's a huge variety of methodologies that those patient groups use when we're collaborating with them. We also work regularly with specialist recruiters or specialist recruitment agencies and patient panels. So they are groups of patients or caregivers who have expressed interest and provided consent to be contacted about research studies that are specific to their disease area or area of interest. Less commonly, we use methods such as social media, so targeted advertising on a variety of different social media platforms, and also snowballing recruitment, which is essentially word of mouth recruitment. So a participant may take part in one of our studies and they may know people through the variety of networks who also suffer from the same condition, and they might pass the information about our study on to somebody else. Each of our studies is different in that we would either use a single method for recruitment or a combination of methods. Now moving on to think about diagnosis validation. So diagnosis validation to us means ensuring that the patients or the participants that take part in our studies are true patients, not just true patients in that they have the diagnosis, the condition that we're interested in, but also that they meet the eligibility criteria for a study. So that might mean things like taking a particular treatment or being in a particular disease stage. And this might sound a very obvious question, but why is it so important to us and why is it so important to our clients? Well, we don't want false participants in our study. We want to be able to show that the data in our study is true to the population of interest, reflects their experience, and we want to be able to provide confidence to our clients that this is what we are providing in our data. Diagnosis validation is relevant to both the quantitative and the qualitative studies that we work on. As you can imagine, it is usually easier to spot somebody who doesn't meet eligibility criteria or is not a true patient during a qualitative study, so during an interview or a focus group. Recruitment and validation is particularly important and worth considering in depth when we are conducting real-world rare disease studies. So as we know, the numbers from these populations, these patient populations, are small. The patients are usually dispersed geographically. And so we want to be able to sample as many of those patients as we can in our studies, but also maintain that confidence that those participants have got that genuine diagnosis and meet those eligibility criteria. And so there's often a balance that a process that we undertake at the start of the study where we may choose less well-validated methods to be able to make sure that we get the increased numbers and to just taking potentially an additional risk. So what sort of considerations are we thinking about? What things are we thinking about when we're choosing our method for diagnosis validation? The choice is dependent on a number of factors, and we'll come on to talk about some of the actual methods that we use shortly. First of all, we think about the design of the study. What does the study look like? How long will that participant be taking part in our study? Is it a cross-sectional study? Is it a longitudinal study? What kind of touch points is that person making? Is the study paper-based? Majority of our studies are electronic digital studies, and so we have the technology within our digital platform to be able to undertake some of the diagnosis validation methods that we're going to talk you through. We also think about the intended audience, who's actually going to see that data from our study and in what format. What kind of scrutiny will we and our clients be put under as to making sure that we can show that we do have true patients in that study? What is the recruitment source? Has that already been defined? For example, if we are going to be recruiting through clinical sites, if our client also wants to collect clinical data alongside the patient-reported data, we may not need to put so much consideration into the diagnosis validation because it can already be done as part of that process through the sites. We would also think about the patient population of interest and consider this on an individual study basis. So by that, we mean things like the eligibility criteria. So what kind of eligibility criteria are going to be defined for the study? Are those criteria things that we can expect participants, whether they're patients or caregivers, to be able to report themselves? We would also consider that patient group as a population. What is their ability like to adhere to the process that we define for validation? Is there anything particular about that group of patients, whether that is something which is mental, which is physical, that may impact their ability to be able to follow that process? As we've already mentioned in relation to rare diseases, we also need to think about the geographic spread of those patients and also the prevalence of the disease. So thinking about diagnosis validation further, thinking about how to choose that method, one of the sources of materials for the workshop outlines 3 different factors that are aligned to successful diagnosis validation. The first one of those is patient knowledge. So if eligibility questions are not understood by patients, they won't be able to self-assess eligibility. There are various ways to help with this, to help to mitigate this issue. For example, ensuring that potential participants have got access to either a specialist recruiter or a member of the study team to be able to help them with any interpretation, any technical aspects, any difficulties that they have in understanding the questions related to the eligibility. There should also be considerations around the cultural differences and levels of engagement, the knowledge around medical conditions. So particularly in countries or in cultures where patients are traditionally more likely to depend on their healthcare professional for information, this may impact the level of engagement and knowledge of that particular population. Moving on to thinking about patient engagement. So some recruitment methods are known to be associated with patients who are potentially less engaged in a study. So the example given in this reference source is patient panels. So patient panels are people who have consented to be contacted about usually quantitative sort of survey-based studies. So they're very used to receiving invitations for potentially numerous surveys, and that can sometimes lead them to not pay full attention to those survey questions, leading therefore to low quality data. In general, in our experience though, patients do want to discuss, share their experiences in our studies, particularly with the hope that they can improve the outcome for future patients. So particularly where we have worked in rare disease communities, we really do see high levels of engagement, enthusiasm from patient groups. And then thirdly, honesty. So this is a principle which really underlies successful diagnosis validation, particularly with some of the methods that we will come on to talk about further, where we're relying on participants to be honest to a certain degree. So some of the methods of recruitment that we've mentioned can be more strongly associated with dishonesty from participants and therefore the enrolment of fraudulent participants. So particularly thinking about those methods that I talked through on an earlier slide, those using snowballing recruitment or open survey access can tend to lend themselves to be open to potentially fraudulent participants. Dishonesty, as many of you can imagine, is often associated with the incentive payment offered or the payment, the thank you payment for taking part in the study. And that can particularly be a problem if that payment is high. So for example, in rare disease populations, we may need to increase payment because there are so few patients out there, or when we are asking participants to do something which will take a fair amount of their time. So for example, sitting through, participating in an hour-long interview, for example, or completing a long survey with many questions. Generally, again, we see that that's more common in quantitative studies rather than qualitative studies. It can be difficult for someone to get through a full 60-minute interview, falsifying information about a disease or a condition. So I'm going to hand over to Sam now just to talk about some of the sources of diagnosis validation.
Yeah, thanks, Catherine. Before we come on to discussing specific strategies for validating diagnosis, so building on what Catherine said, we thought we'd just sort of highlight, quite high level, some of the most common sources of diagnosis validation, and sort of just outline some of the advantages and disadvantages of both. Aware of the fact that many of the people attending the webinar today as well will have their own experiences, good or bad, with all of these. So, of course, the first one there, and Catherine touched on this as well, was patients or study participants recruited through clinical sites. So obviously a benefit of this is that we're able to, accurately confirm the diagnosis from clinicians that we're working with at these clinical sites. And also listed here is that, again, through working with clinical sites, we would typically expect that we're able to get more information about a diagnosis than potentially a patient. Also thinking here, with that, if it's a study involving minors, again, or a rare disease where potentially the study participant isn't as familiar with all of the aspects of the disease itself that is required for that study. So those are some things that clinical staff are able to provide in addition to a participant there. Working through clinical sites specifically, then obviously a disadvantage is that it can be both expensive and time consuming. So again, I'm sure a number of people on the webinar today have worked or tried to work with clinical sites to recruit participants for their studies and have similar experiences of that. So something worth considering if, and again coming back to sort of what Catherine was just saying on the last couple of slides, if you wanted to go down that route of diagnosis validation, just to bear that in mind, that you would typically need to allow a lot more time and budget to obtain that kind of diagnosis. Linked to that then is receiving confirmation of diagnosis from a clinician by way of the participant. So this could be, as part of registration, there is an expectation that a study participant will get in touch with one of their healthcare professionals and obtain, some sort of, some sort of confirmation of diagnosis, whether this is a diagnosis letter or, a bespoke letter could, for example, be created for the study that, the expectation is that they then get signed off by a treating healthcare professional. So again, similar advantages, accurately confirmed, if that's e-signed or stamped, that, we can, those looking then at the validation as part of that study are able to confirm that. And again, if, for example, a bespoke confirmation of diagnosis letter was created for the study, we could, when deciding what level of diagnosis information was required for that study, actually request more information as part of this letter for the participant to then take to that healthcare professional. Disadvantages, sorry, again of this are, and we will come on to this a bit more in the next slides, but it's an extra piece of work required of the participants there. And typically, again, just coming back to sort of time and budget, both of those, additional of both of those would be required if opting for this source of diagnosis validation. We can't expect participants to just be able to, email or pop in and visit a healthcare professional at short notice to be able to get this confirmation of diagnosis. Typically, some more time is allowed there. And again, compensation. So, there would typically be an expectation for participants being required to go out and get this confirmation to receive a slightly higher compensation, perhaps, than on a study where we weren't opting to use this. And linked to that is obviously that risk then that prospective participants joining a study and seeing that there's this additional requirement may actually be put off by it. We can't assume that all study participants or prospective study participants are comfortable necessarily reaching out to the healthcare professional and obtaining this. So it's an extra barrier there that's worth considering. And similarly linked to the sort of time that's required to that is the variable response rate. Healthcare professionals are extremely busy. It seems now more so in the past couple of years than ever that actually it's quite difficult to get in touch and to get in touch with healthcare professionals, and we've seen that. We've had anecdotal feedback from participants in our rare disease studies that even healthcare professionals specializing in the rare disease that they have are still being pulled in different directions, and actually that could lead to an extra barrier there. And if we go to the next slide, Catherine. And then another common source then of the diagnosis validation is to get that confirmation of diagnosis from the patient, so self-reported. An advantage then over the two other sources that we just went through are that that's quick and easy. Another advantage sort of linked to back on a previous slide, Catherine sort of mentioned how we would typically, the source of participants for studies, and one of those being, working closely with patient associations, patient advocacy groups, and actually, where we're recruiting through that method, participants, or prospective participants rather, would typically be quite knowledgeable of their condition. So I think, it is an advantage that if that method of recruitment was selected and then there was a requirement to, self-report their diagnosis, then I think, it's definitely, better and more reliable than if we were, going back to Catherine's example, if we were sort of expecting that from sort of the snowballing technique, for example. Disadvantages of this source versus those covered on the previous slides. Again, fraudulent patients, always a risk. If we're just asking participants to sort of answer a few questions to confirm their diagnosis, there is that risk that someone might quite quickly check all the boxes, sign something themselves. So that, again, it's definitely a risk. And again, sort of linked to where then there's advantages of working with clinical sites and healthcare professionals directly to validate that diagnosis. Patients or caregivers might not be able to accurately report as much information about the diagnosis as we might want for the study. So again, that's just something that needs to be considered as we think during the design phase of a study about the source of validation that we want. And if we could go to the next slide, Catherine. And I think back to you.
So we're just going to take you now through some of the strategies that we use commonly for validating diagnosis. So the first one is monitoring the data on an ongoing basis. So the data that is coming into the study can be monitored almost in real time with the aim to filter out any unusual response patterns. So in our experience, unusual response patterns can potentially indicate fraudulent participation. So those people who move through surveys very quickly, so spend a lot less time on questions than we would expect them to typically. Another example could be those who provide the same response options repeatedly for a number of questions in a row. We've also had examples where people have given unusual responses to free text options when we're asking them, for example, for the name of the medication that they're taking. So we're able to pick those up very quickly and then filter that data out of the study population. So one of the advantages of our data collection platform is that the data is reported from the study in real time. So we're able to very quickly identify potentially fraudulent participants and to remove that data. We would also always include screening questions as part of enrollment. So whether that's patient-facing screening questions or screening questions for clinical sites to address. The idea being that the questions are phrased in a way that only a true patient with the disease should be able to accurately respond to. And we would ensure this or maximize understanding by testing those questions with that patient group first. So it's really important that they are phrased in a way that is clear and that's well understood. And we'll go on to giving a couple of examples, and we'll talk you through how we would typically phrase those questions to be able to filter out the participants that we're interested in, those that are able to confirm their own diagnosis. As we've mentioned, we commonly recruit through patient associations and support groups. The pool of those participants are much more highly likely to be genuine. As we've mentioned, they're people who have got an interest in engaging with others with the same condition. Again, we will talk about this when we come on the next slide to take you through a couple of real life examples, but we have also used several situations where we've requested potential participants to scan or upload proof of diagnosis. So that could be a letter, it could be medication packaging as part of that enrolment process. So generally it's easier for potential participants to use a smartphone, just to take a photograph of their medication packaging and to upload that to us. But there are, of course, situations where that might not be applicable or appropriate. So if the treatment of interest is administered in a hospital setting, for example, the patient may not have access to that medication packaging, and we might need to consider things like uploading letters of diagnosis confirmation. There are also some technical aspects to think about, so things like the clarity of the image, how we would extract the medication name, and the sort of more technical components. We would always advise using open methods of recruitment with caution, things such as social media advertising. These things are very much used as secondary methods in our studies, very rarely put forward as primary methods of recruitment. So just to take you through a couple of examples, so I'll take you through the first example and then Sam will talk you through the second. So the first is using screening questions as part of enrolment, so making sure that those screening questions are understood by potential participants, and also that they are able to filter out participants who don't meet the eligibility criteria. So an example here, if we were interested in patients, for example, with stage 2 bowel cancer, rather than ask them, do you have stage 2 bowel cancer currently, yes or no, we would list out the stages. And so we're much more likely to rule out or eliminate anybody who picks an answer at random. And we then may go on to have a series of questions that are phrased in that way with open response options, rather than those which are yes and no. So, for example, we may then go on to have a list of treatments. We may ask that potential participant which treatment they're currently taking, if any. The second example from one of our studies, the questions here are in French, and they're an example of where it has been appropriate to ask patients to classify their own disease stage. So this is done in conjunction with clinicians who agreed that this was an appropriate thing to ask this group of patients to do. And so we developed some language in collaboration with the clinicians to describe the different disease stages, and so that patients were able to classify themselves according to the stage which they felt that they were most closely aligned to. An example where that's particularly applicable would be if the disease stages were characterized by levels of mobility. So patients are able to tell us about what they are able or not able to do physically. So that's a good example of where we would be able to accurately ask them to classify which stage of disease they're at. But as we said earlier, these questions are formed, discussed, considered on an individual project basis. Sam?
Yeah, thanks, Catherine. So another then of the strategies for validating diagnosis that we've implemented in a few of our recent digital real-world evidence studies and are very excited about as well, is the sort of upload feature. So again, where these are sort of digital, obviously, remote longitudinal studies, and sort of circumventing the problems then of someone having to print something off and take it in to a healthcare professional is actually, if they're able to get this letter somehow or have an existing letter, actually allowing them to scan this in or take the photo while they're using the app and scan that then as part of registration. So the screenshot there might be a bit small for people, but essentially what we've implemented in a study recently is asking participants as part of registration to upload a copy of their genetic report. So this is, again, coming back to sort of strategies of why we're doing this, and it's sort of linked to this to the screening questions as well, as that participants are asked and aware as part of enrollment to the study that they will need to provide a copy of their genetic report in order to participate in the study. So again, eliminating that risk of fraudulent patients is that a fraudulent patient wouldn't be able to do this, so they wouldn't or would be very unlikely to be able to do this, so they would be unlikely to proceed into the study on that basis. What I would add then on this is obviously that there's extra considerations to be had then around data privacy. So quite typical on these sort of longitudinal digital studies is the need to get app terms of use and privacy, data privacy policies, really tight and detailed and clear to the participants. So, for studies where we've implemented this reward upload functionality, is that we've been very clear in the privacy policy about sort of why this is being done, what's being done with the genetic reports then once they're uploaded into the database, and actually part of that, and this comes to the second sort of use of this kind of functionality, is that something else we could do is ask participants to upload scans of their medication bottles as another way of validating diagnosis. And actually what software, including our own, can do is actually review images and just pick out keywords. So, these can be defined up front. So also thinking about the genetic report, if there was sort of key genes that would typically be listed on a genetic report in this patient group, having the software actually read those and redact everything else. So it actually, negates the need for someone to manually sit and remove any personally identifiable information. And again, making that really clear to participants up front as well sort of really helps, calm a lot of concerns that we see in these types of digital studies, which is, what are you going to do with this this information about my diagnosis once you obtain it? And yeah, speaking to participants about this feature and expectation, anecdotally at least, has been quite positive. So yeah, that was that one.
Thanks, Sam.
Thanks both. So now we're going to move to the Q&A section. I've got a few questions here that I think touch on actually a lot of what you've just said just now in the examples. But one is about not just on how the diagnosis is validated, but also whether or how the severity grade is validated. So, for example, PSSI score or MGFA grade for MG. So I don't know who wants to take that.
Sam, do you want to take that?
Yeah, so I can take that one. So I guess, yeah, on that question, Anna, so yeah, sort of touched, I guess, later on in the webinar was the example then that Catherine took us through. So actually, we worked quite closely with clinicians as part of the design on that study, who formed part of an advisory board to come up with the screening question, which asked participants to essentially, self classify themselves. And actually part of that, so admittedly the example was in French, but what we had there was a standard classification system. And while a participant might not necessarily know which class they're in or remember from any conversations they've had with healthcare professionals, is actually, if we can provide a sort of patient-friendly, patient-centric description of each of these classes and have those descriptions validated by clinicians, who are experts in this field, is actually then a way that we can do that. So a participant would not necessarily, know the class off the top of the head and be able to respond if we just said, which class are you, 1 to 5. Actually having a description, they're able to sort of rate themselves, but in a reliable way, in that the descriptions are sort of, what we would expect then clinicians to be using themselves. And that can be done in various ways. I'm sure, depending on the condition or the symptom, I guess, that we're looking to have classified as part of enrollment into the study.
Yeah. Okay.
Just to add to that, the example that was mentioned in the question that came in was the PASI score for psoriasis. So there are some situations, and this is an example of one, where there are validated patient forms of severity measures. So there's the self-PASI for patients to complete. So we would also always consider whether these are appropriate to use in those individual situations. So it may be that something has already been designed with patients in mind.
Fantastic. Oh, thanks both. I've got another question for you here. So do you have to pay patients extra to seek confirmation of diagnosis from a healthcare professional?
I can answer that one. So yes, I mean, you don't have to, but we typically do. We would typically compensate patients or participants on the time that they are participating in the study. So whether that's an estimate of how long we think it would take them to complete a survey, or the time that they will spend in an interview or a focus group. And so we would also need to consider the additional time that we think it would take somebody to be able to undertake whatever activity we define to be able to confirm the diagnosis. So we anticipate that would involve extra time, extra effort, and so we would advise compensating the participant for that. And there may also be situations where we would also advise compensating the healthcare professional as well.
I guess just linked to that, Catherine, as well, is that that doing that isn't necessarily to. So the asking for the confirmation of diagnosis there isn't necessarily to put off fraudulent patients, but actually it's so that we end up with a cohort in our study who we have, regardless of whether they've been paid or not, but we are confident that they are participants with that disease. And so then it's not just about the short term, what you're doing in the study and the data set. It's actually thinking about long term, these longitudinal studies. Who do we want in the study? We want genuine participants that are going to be engaged and providing data, but then also thinking ahead to dissemination, publications, dossiers and stuff, actually being able to include then in a manuscript, presenting results, is that we obtained confirmation of diagnosis, being quite clear in that sense. And actually, so it's not just short-term benefits of who we get into the study, but it's also those long-term benefits, rather than those questions coming up 3 years into the study and us having not considered them at all. I think it's worth doing that up front, and at least if you don't include and you don't offer that extra compensation for obtaining this confirmation of diagnosis, having a rationale for why you're not doing that, and maybe using some of the other strategies that we've touched upon.
Absolutely.
Actually, because you've touched on the potential for fraudulent participants, there's a question here that's sort of made me smile, but how often do you identify false participants in your real world studies?
Yeah, we can go. So we're laughing as if we're covering something up, but no.
Yeah, I know.
No, it's very rarely, and I think it's testament to how we at Vitaccess anyway would typically recruit participants. So I know you mentioned a few times, Catherine, and it's, you know, in line with my experiences, is that we would typically work quite closely with patient associations. So again, you know, we can be quite confident going into those relationships and into recruitment, knowing that actually the participants who are going to be exposed and spoken to about this project through people that we're working with, or we're able to get in touch with them ourselves via you know, various channels, you know, we can be quite confident going in that those are genuine participants. You know, and, you know, working down, I guess, priority of recruitment channels, again, working quite closely with you know, trusted participant recruitment agencies. Again, you know, there's a level of expectation going into that, and trust, sorry, not just expectation, that actually the participants who are part of these agencies cohorts from which, you know, recruitment can be done, you know, those are genuine participants, engaged participants who have been there for a long time, rather than, you know, a fraudulent participant, say, that sees a study advertised and there's mention of a reward, and then they suddenly, you know, work their way into a patient group at short notice. I think that would be quite difficult. So yeah.
Yeah, it's clear that there are like multiple ways to make sure that, you know, the false participants are not becoming part of the study. We're running low on time, so I'm going to ask one last question. If you use a combination of recruitment methods, do you start them all at once or do you stagger them?
I can start on this one if you like. So I think it really depends on the study. So we've got examples of both. Each of our studies up front, we would always put a recruitment plan in place. So the recruitment plan may outline starting with one or just using one single method of recruitment throughout that study, starting and ending with that. It may involve using multiple methods and again running those from start to finish, or it may involve looking at patient numbers in a real-time basis throughout the course of that study and then introducing methods depending on the success of each of the previous methods. So for example, we may define a minimum number of patients that we want to get from social media advertising, for example, and then if we don't hit that minimum number by a certain date, we then may think about introducing another recruitment method. But it really is individual. Our studies are so varied. If we're working with clinical sites, we usually tend to work just with clinical sites because they're able to conduct some really in-depth feasibility for us to work out the number of participants that we're able to get that match the inclusion criteria that we need to, and that those patients are the eligibility criteria. So it really is individual, and we use both of those approaches.
Great. Yeah, so it depends, isn't it? Really?
Absolutely.
Yeah. Fabulous. Okay, well, that's all for the Q&A. So our next webinar in the series is on how to avoid obstacles and barriers to RWE analytics. And this is at 2 p.m. British summertime on the 21st of June. So it's the first day of summer for anyone on the call who's in the Northern Hemisphere. And it's led by our data analytics experts, Dr. Casey Quinn and Amber Kudlak. So this one's a really good one for those of you who are starting on designing your own real world study, or struggling to get the most out of your data as well. So the link to register, I'm going to drop into the chat here. So please feel free to sign up. And that's all for today. And we hope to see you next time. Thanks, everyone. Thanks, Catherine, and thanks, Sam.
Thanks all.
Thanks, everyone.
Bye-bye.